Now they're in
Yikes.
that hole on the right.
I'm at 36.
Thermocouple temperatures at 24.
43, 48, 49, 53, 54, 59, 65, 68, 70.
A good feeling about this one. Finally.
Thermocouple temperatures at 21. But I expect
It's
that
back
to be lower.
60.
Nice.
Are you okay with this?
You're the
Or
boss.
is it too high? But these are your guys.
What?
Your babies.
The Cillians?
Yeah.
They're my babies?
Yes.
Would you guys consider this diffuse? Seems kind of pinpoint to me.
It's more of a focused flow,
Yeah.
for sure, but it is a lower temperature one. How do you feel about that, Amy?
Do you
I think
concur?
it's okay. I mean,
Okay.
What's the temperature read on?
Now it's 53.
yeah, it's between like 43 and 63.
That's probably what you want, right?
Yeah.
It's locked in, Trevor?
Can't hear you.
It's locked in. Yeah.
Yeah, it's locked in, yeah.
I just have a quick question. Are you right aside the temperature that has been measured at 113?
Could you repeat that and speak up a little bit?
I'm wondering if the temperature that we are looking at right now is just like a dilution of a fluid that is actually at quite higher temperature right below the few centimeters where we are above the surface, and it's just the lower temperature just reflects late dilution with seawater, if
Yes.
you see what I mean.
Yes, I think so. And that's kind of what we saw the other day when we were at that at the colonization chamber area, and when we took the diffuse flow measurements. Is it seemed like that the mixing at the surface here, as the hot seawater is coming out, is when we're getting the lower temperature. Do you want something that's coming out at a lower temperature?
No, I'm okay with that. It's just, yeah, because we measured the temperature that was quite close to the limit of, let's say, 120.
Yeah.
Yeah, I was wondering if it was not maybe too hot, and even if we can measure lower temperature, if it just reflects really late mixing with seawater, maybe, I don't know, there won't be much microorganism in it, or I don't know exactly, but yeah.
Yeah,
just
I mean,
wanted
I have the same kind of concern, but it seems like we just keep finding those areas where it's really hot fluid coming out no matter what, and it depends on how much mixing there is, and then like if it's really focused or not, as to how much or what temperature you get it, and within like a centimeter, you know, it varies quite wildly, so it's really difficult to really get a handle on if there are microbes in there. Are they just living at the surface, or are they deeper down? It's hard to say.
Okay, good. That's fine with me.
Okay.
Thank you.
So we're at 24 degrees, Trevor, so can we try to get to a little above 50?
Absolutely.
Okay. $30, $34, $37, $45, $47, $50, okay. $55, $56, $62, $55, $42. Yeah. It's just the movement with seawaters. 51.
It's going to go back up now.
Yeah, it's just going to go up and down. So I think we're good to go. 67, 69. It's going to be really difficult to keep it steady. Are you okay with this?
What do you think
number?
the temperature range is going to be for? Do you think it's? Should we watch it for another few minutes?
Yeah, we
seconds?
can. Right now, it looks steady in the 60s.
I bet it's cooling off soon.
You see how that starts wafting away?
Yeah.
It'll change the temperature.
Now it's 52, 49. 44.
Yeah, just the subtlest flow change in the water.
Yeah.
Changes it extremely. I guess you'd solve that problem by getting the wand closer, but then it's too hot.
Right. So it'll be like between 45 and 65. Is that okay with you?
I think that would be fine.
Okay.
It's still going to be in that higher temperature range that we haven't gotten yet for
Yeah.
the diffuse of the in that range.
Okay.
We'll watch it.
Yeah. So I'm gonna go ahead and start sampling here, Trevor.
Roger that.
We're doing filters. We have the camera on the flow meter.
Just getting set up here. Okay, we're standing by.
So this is going to be filter number 4, S4.
That is correct. I'm going to port 4. Whoops, I need to be on the screen. Now I'm going to port 4. Okay. And I'm going to be filtering for 20 minutes. And I'll do that for 3 filters. Okay, Trevor? And then we'll...
Roger, yep.
And then we'll switch the wand and do 1 bag.
Okay.
Okay. And start filter.
Copy that, starting filter.
And if you see the temperature drop too much, we can reposition the wand or whatever.
Okay. I see it's at 37.
And just for reference, the thermocouple is been between 12 and 17 degrees C. Again, we know that that's going to be different from the super because it's outside of the flow.
Oh, I don't think this started.
What's the matter?
It's
There's
just
a
going
flow
really
rate.
slow.
It's, yeah, it looks like
the
it's,
flow meter's not going. Oh, now it's starting to go.
yeah, it has a bit of a lag, I think, doesn't it? For this,
Mm-hmm.
does it have a slow start?
Come on.
It's not quite going yet.
I don't know why it's having so much trouble. What do you think, Eric?
Well, it's definitely filters, because it was moving through the purge positions fine. It's moving a little more now.
Yeah.
Might be getting the air out of it.
It's starting to go now.
Yeah, I think there was air in it.
Just note that it took about 1 minute and 45 seconds
To really start
to really
moving?
start going. If you
Okay.
want to start the time now more.
Start the time now more.
You know what I meant.
You mean start from this time and then
Yeah,
do
or just
20
add
minutes from here?
add a minute
Okay.
and a half.
Okay.
Yeah, it looks like you got a better speed on your flow meter now.
Yeah.
Yeah.
It's more consistent.
It's going faster now than it was on the other filters too.
Is the temperature holding?
Yeah, it's about 32. It's been steady in the 30s. Now it's up to 41. 43.
Did you guys see Chris's comment on side chat?
We're not on side chat.
He says he's still concerned it is not a problem that the fluid coming out of the ground might be over 130 C as it exits the seafloor, question mark. So the fluid being sampled isn't too hot, but the water coming out of the ground here would have already been sterile?
Yeah, I don't know.
I felt like most of the flows out of this area were pretty high,
Yeah, they are.
but we still see growth on the outside.
Yeah.
Vincent says, please can you keep writing the temperature in the log time to time?
Copy that, Vincent. Yeah, our data logger is continuously monitoring the temperature. So when Amy and Sarah call out both the super and the ICL respectively, Aubrey will be popping it into the data log for you guys to keep track of. Yeah. So you'll be able to hear it verbally from the guys in the van and then also digitally as well as it comes through the data logger.
I'm back up to 53.
The ICL is at 14, 15.
Can we get a quick temperature update, please?
Sorry, 32.
That's okay.
ICL is at 10.
Great. I know you guys are focusing on screens and stuff, so I'll just
I was
prompt you every
asleep.
I'll prompt you every minute or so, or every
Yeah.
couple of minutes.
I was just thinking about the flow. Even though there is hot focus flow coming out right there, and it might be up to 130 or so, that whole area and that whole crust could be at a much lower temperature, let's say 90 to or 70 or something, and that's probably also mixing in there. And just if the microbes are coming in contact with that hot fluid for just a minute or two, it's not going to kill them. It just over time it will denature their protein, so that's why they can't grow over 120. But I think we would still see some microbes in the fluids.
Cool.
Yeah.
Yeah. No, if you guys are happy, we're happy. That's good.
I think so.
As you say, it's really difficult to tie it down, especially when the fluctuations in
Yeah.
the temperature vary so much.
Yeah.
With the entrainment and then
If
the
this
position
was
of
a
the-
big chimney with a giant, focused flow, I would, think differently. But because this is more of a larger area with a little bit of flow
Lots of
coming
little
out here and
fumerals.
there,
Yeah.
I feel like it's probably be okay. Yeah.
Good. Excellent.
Is it an okay time for me to ask a pedantic technology question?
Okay.
All right.
I'm assuming that means it's okay. Is this the IGT temperature sensor? Is it a thermocouple or is it an RTD? Because I know our temperature probe, the vehicle temperature probe, is not a thermocouple. It's a RTD.
I believe Sean's is a thermocouple. Mine's a RTD with a platinum resistor on it.
Are you, sorry?
Mine's the one in the Super, that's built in the Super, is RTD. It's got a platinum PT100 on it.
Okay.
And then I think Sean's is a thermocouple.
Okay.
And now for those of us who don't understand what the difference is between those two.
One measures...
the PT100 measures resistance. It's got a reference resistor as well. So the difference between the reference resistance and what it's come back as, that's how you measure the temperature.
That's RTD is the acronym.
That's the RTD, yeah.
And what does that stand for? Resistance temperature difference?
Resistance temperature detection.
Okay.
And what is the resistance in? Is it the material or?
It's at the end, it's a resistor. It's a platinum resistor.
That changes with the different temperature.
Yeah.
Okay.
And then thermocouple is two dissimilar metals that produce a voltage. Being dissimilar, all dissimilar metals touching each other produce a voltage. If you know that know the dissimilar metals touching, you know what the voltage is, and that voltage changes with temperature. So it's calibrated with not a lookup table, but like a linear-ish change in temperature, changes that voltage created between the two metals.
Interesting.
Because of the change in resistance, though?
Not in a thermocouple.
Not in a thermocouple.
Can we get another temperature reading?
They want another temperature reading.
Oh, sorry. I'm getting tired. 34.
ICL is at 9.
Great, thanks guys.
I had too many naps yesterday, and I didn't sleep last night because.
Oh no.
You were too excited?
I was going to say.
Yeah.
Excited for the dive.
Too much rest.
Yeah, this is a long one for you guys as well.
Yeah.
But full of microbes. Your flow meter seems to be moving quite consistently now, actually. It's got a good speed on it. It's really consistent.
What, Sarah?
When we're doing the next filter, go get some coffee if you want.
Oh.
20 minutes. Or I can go get it for you.
Okay. I don't want any coffee.
All right.
I might have some water, though.
There's some espresso beans there, Amy.
What?
Huh?
There's espresso beans.
Oh.
Okay.
That means yes.
Oh, it's like chocolate.
Is it a dark chocolate?
Ish.
Snickers.
It's not super sweet.
Okay. I'll have one. Oh, shh.
There it goes.
So
Bean?
Yeah.
someone would like to know who keeps an eye on Argus. If Argus is watching Hercules, who keeps an eye on Argus? There's an Argus pilot here.
Yeah, there's an Argus pilot, which is me.
Mm-hmm. Hi.
And who is me? Introduce yourself.
Tell us about what you do.
Hi, my name is Cassidy, and I'm the ROV intern. So this is my first cruise. My job is to make sure that Argus is keeping an eye on Herc, basically, and keeping the tether, in sight and, not tangled.
Can we have a quick temperature? Oh no, stand by.
Looks like 32 degrees.
On the which sensor?
Super.
Super temp is 30.
And ICL when you're ready, Sarah?
13.
13, thank you.
I can see that.
Someone's mic's hot. I don't know if someone's got a...
Turn your mic off. Yeah, remember if you guys are moving your mic to mute yourself before you do. It creates a crackly, poppy sound on the audio.
I'm at 41. And the super's at 10.
Super is at 10?
Yeah. I'm sorry, the thermocouple.
Okay.
Do we have a estimated flow rate for the sample too?
I don't know.
We're off the coast of Northern
Sure,
California, Oregon, at Gorda Ridge, collecting samples for NASA subsea scientists, Ocean Exploration Trust, and Woods Hole Oceanographic Institute, and NOAA, as they explore the hydrothermal vents and look at geological and microbiological organisms as well. Matthew, could you possibly explain how this is serving as an analog for studying space exploration?
We don't actually have Matthew or Zara in the van at the moment, I'm afraid.
Okay.
I don't know if they're in their lounge and they want to be able to, they can contribute from there.
Hey Data, can you tell me when this sample started?
2116.
2116. Copy.
Eric, can you tell a flow that's gone through the filter at this point?
Did she give you starting flow counts?
No, she didn't.
Okay, wait, I see here. Just give me 1 second. It's like 15 minutes that passed. The flow rate's about 0.37 liters per minute so far.
Thank you.
And you said it started at 2116, correct?
Yes, 2116.
Eric was commenting earlier how little the sea spiders move. And they don't move a whole lot. I've been watching them. They don't move.
How many organisms can we spot in that screen at one time?
Let's start counting. We might
like
get
different kinds,
oh,
we'll
different
say.
kinds.
Not individuals would take too long.
So, yeah, we got pycnogonids. We got, I think, 2 types of gastropod. I think there's limpets, and then there's another smaller type of gastropod there as well. That's some scale worms. Some sneaky para-alvanella. Can you see the plume to the right-hand side? Just in that little crack there. There's a few para-alvanella in there. And then some of the Rigidiopisci as well. It's quite good, actually. It's quite representative of the fauna that can be found at this site.
Yeah.
We're seeing quite a lot of it here, and that's probably owing to the fact that we're actually in a lower temperature area. It's not dominated by, even though we have this focus flow just in front of us, actually the rest of it's experienced more probably a lower temperature, hence the number of animals, or the different diversity of them.
Can we get another temperature reading?
Yeah, temperature 34 on the super.
Hey, it's Darlene.
SPL.
Amy just flagged me down to say that somebody wanted a comment about analogs. Is that true? Over.
Yes, that's true. If you could explain to our audience the importance of this. They wanted to know, people had questions. as to what was the purpose of this and what are you learning from it.
Okay, so let's back up a little bit. What is an analog? An analog is an environment or an operational or some sort of operational setting that offers us a point of comparison to other environments or operational settings in our solar system. So what I mean by that is that we can look to our Earth and to different settings, whether it's deep under, the ocean in this case, or let's say to a volcano, or up to the Arctic or the Antarctic, or down to the Antarctic, and examine different environments as they may pertain to understanding life on our planet, as well as on other planets potentially in our solar system. And then we can also use these environments to learn about humans and robots and software and different types of hardware and so forth, and how they might eventually support human exploration in our solar system as well, and how do they perform in these environments that are pretty close approximations, we think, in some way, shape, or form to somewhere else in our solar system. So that's what an analog is, and that's what this is as well for NASA, this being subsea. We've got 3 different research pathways to our subsea program. One is the natural sciences, the other is the social sciences, and the other is technology. And all together in an integrated way, they are serving as a NASA analog under the banner of subsea. And there's lots more details, and you can search that out on our website. If you just Google NASA subsea, you'll be able to see a lot of information there, as well as on the nautiluslive.org website. There are expedition pages that you can read through that give you a lot more detail. But we're kind of examining this deep sea hydrothermal system as an opportunity to potentially examine and look at this environment through the lens of looking outwards in our solar system, as well as looking at the way that we do business on the ship and on shore in terms of the science communication, and how that might be a possible analog to how we will eventually communicate when we have humans exploring in space deeper and deeper into the solar system. Over.
Thank you.
Getting 45 degrees. Can you hear me on here?
Copy that. 45.
ICL is at 13.
13. And are we overrunning by a couple of minutes to account for the slow start with the flow meter?
Yeah, we'll stop it in about 30, 40 seconds.
Copy that. Standing by.
So someone would like to know what's the source of the methane and hydrogen sulfide in the vent fluids?
Real quick before they answer that question.
Sure.
When the sample's over, if we can just get a flow rate and a total volume, that'd be great.
Maybe we can answer that question in a minute, Susan.
Did you stop
absolutely.
it? Are you stopping the flow?
Yeah, I think I'm gonna stop at 38.
Okay, standing by.
Stopping sample now.
Copy that. Stopping sample. 2138.
End flow count is 10087.
10087. End flow count.
Total volume was 7.5 liters.
7.5 liters. And the rate?
And the rate was 0.38 liters per minute.
0.38 liters per minute. Copy that. So total volume 7.5 at a rate of 0.38 liters per minute.
Sorry, the next filter?
Switching ports now to port 5, S5, that is.
S5, port 5. Copy that.
We'll start at 2139.30.
Standing by.
Starting sample.
Copy that. Starting sample 2139.
Confirmation on the flow meter.
See if it does the same thing it did last time.
It started fast, now it's going a little slow. It's probably getting the air out of it.
Yeah.
Yeah, do you think the initial one is just to push some air through the filter?
Yeah.
Probably.
Struggles. Your start count is the same as your last end count, 10087, was that correct?
Correct.
Copy that.
Can I get some temperature readings too?
Yeah, great.
Got 35 on the super.
12 ICL.
So I think we had a question, Susan, was that correct, about the source of the hydrogen sulfide or methane within the vent fluid?
Yes, exactly, and the methane.
Okay, I might actually defer that to someone at the ISC if they're willing to answer, or one of our geochemists.
Absolutely.
So if anybody at ISC would like to take that question. So the question was, where is the source of hydrogen sulfide and methane coming from at these vent sites, and do we know it, or how would we tell?
I'm getting a temperature of 70 right now.
ICL is at 16.
7016.
Correct.
Thank you. I think we said this the other day as well with the para-alvanella, they actually react to the fluid flow as it changes, so they've all come out now, you can see the plumes, and then all of a sudden they all retract. So I think they do change their behavior based on what the fluid flow is doing.
Data 45 degrees and 3 degrees.
Thank you.
Okay, we're aboard the EV EV Nautilus off the coast of Northern California and Oregon at Gorda Ridge, collecting samples from the hydrothermal vents with subsea scientists, Woods Hole Oceanographic Institute, Ocean Exploration Trust, and NOAA scientists. Amy, did you have to have special bags when you were collecting samples the other day so that upon the ascent they don't rupture?
Amy's not here right now. I took over.
Okay,
what was
you
the
can.
question?
But we did have bags specially made for this that fit inside those boxes. They're actually bags that hold wine as well. That's kind of how that's their other use. So they're not gas permeable.
Oh, okay.
Over short amounts of time, yes.
Yes.
So that's why it's really important for us to understand the flow rate as well, because especially when we're filling a known volume, you don't really want to overfill, right? That's correct.
Exactly.
So it doesn't matter so much with the filters, because you can filter over and then calculate how much water has actually gone over the filter itself. But with the bags, they're of a known volume, so we want to be a little bit conservative that we don't overfill.
Correct. We don't want any bags to explode.
No, not ideal.
No, absolutely not.
Exploding no good.
Right.
Can we get a quick temperature update when you guys are ready?
20 degrees.
ICL is at 26.
So we swap positions.
Yeah. Well, look at the flow. You can see
Yeah.
the flow has moved a little bit.
It's flown to the right more so.
Yeah, the ICL is now at 39, 40.
It's certainly shifted, hasn't it, over towards the right?
Yeah.
Or the current shifted.
You want me to pause the flow at all?
I don't think so.
Okay.
How will these be grown, these microbes?
We have different media types that we use in the lab where have previously shown to grow microbes from other deep sea environments. So it's a little bit of trial and error.
Mm-hmm.
Amy, that's what's one of the projects Amy's working on while we're here. So she has a variety of different media, and she'll be inoculating some of this fluid into those media types.
Okay, thank you.
Are they the same media types that you're using in your colonization experiments, or are they different?
They're different. Some of the minerals are the same, I believe, that Amy's using. But the microcolonizers have bits of shell, quartz, olivine, and I believe basalt.
Okay, cool. Sorry, I just interrupted your temperature reading as well. Do you want an ICL update?
Is the temperature back up on the super?
Yeah.
Yeah. ICL is at 14.
Thank you.
Yeah, the fluid's back to where it was flowing before.
Are there special enzymes or lipids that you find in these hydrothermal vent microbes compared to elsewhere?
Compared to other hydrothermal vent systems?
Mm-hmm.
We have not. No samples, to my knowledge, for microbiology have been taken from these sites to actually look at the enzymes and lipids. So that's something that we can start to address soon.
So this is cutting edge. Mm-hmm.
I just had a temperature spike up to 70, but it's dropping again.
ICL's at 15.
Copy that, thank you. So these, I've got a question about ciliates, Sarah, and I don't know if you can answer it. So I'm just looking at the like morphology. So though they're colonial, do you think this kind of globular kind of consistency or texture we're seeing, is that like, is that what they look like typically? I don't know. I think from a distance I assume them to be more mat-like, but now we're staring at them, they look very globular. Is that what you would expect, or is that something you've seen before? Or
I've actually not seen them before, other than, you know, snapshots from other videos and cruises. So I'm not sure how they're going to look when we first approach them. I think it'd be more obvious if we pick up some rocks with them.
Yeah. Because the stuff we picked up on a rock the other day definitely looked more like a film than this more globular.
Yeah, correct.
But also it was a different color,
Yeah.
right? It was kind of pink rather than this blue-purple color.
Yeah, I don't have a good sense of, right, they're going to have a chitinous kind of tube, and they're going to be inside and probably emerging from time to time, kind of like the tube worms, but I don't have a good sense of size.
No.
Right?
I think that's it. I think I've just started to think about how small these things are. And they're really, really tiny, like the actual ciliates themselves. Like,
they're on the larger size for ciliates.
Okay.
But they're going to be smaller than some of the other organisms that we see right now.
Yeah.
So yeah, I'm not entirely sure.
It'll be interesting to get them back in the lab and have a look.
Does anyone know if tube worms are closely related to porifera, like to sponges?
No, they're two different phyla.
Okay.
So your tube worms are actually polychaetes. So they're in the family they're in the phylum Annelids or Annelida.
Oh, okay.
Porifera are sponges, and that's a totally separate phyla. So yeah, they're very different.
So porifera would be much more ancient.
Potentially, yes.
Mm-hmm.
And they're, yes, as I say, they're sponges and not segmented worms.
Mm-hmm.
When they first found these type of tube worms, didn't they propose that it would be like a whole different phyla, this Pogonophoran phyla?
They did indeed, yeah, they did. They thought Pogonophora was what they were called originally, and then they got reclassified. So they, I believe now, well, these guys are cyberglinids, cyberglinids, and then I don't know where Vestimatifera now sit in terms of taxonomic classifications. I'm a little bit rusty. But yeah, the Pogonophora, they were once called that, and then they've actually...
It's impossible not to be rusty with those things.
Sorry?
I said they change so fast, it's impossible not to be rusty with those things.
It is, and if you're working across multiple phyla in different systems, different depths, it's, and as you say, taxonomy changes a little bit. A lot. A little bit a lot.
A little bit a lot, definitely.
Depending on who you talk to.
Yeah, that's true, depending on who you talk to. And how many samples they have and how many opportunities they have to describe these things. And then when they get the DNA and the genetics, it all changes again. So.
I know, I find that part so interesting. The molecular approach to speciation. It's like,
Absolutely.
What's the precision limit, you know?
So are these ancient, like with the corals and the sponges, or are they fairly new tube worms?
That I don't know the answer to in terms of like, as and when they all branched off. I can tell you in kind of an evolutionary history to all the phyla. I wouldn't want to misinform people.
I'm just curious.
Data, I got 30 degrees.
ICL's at 15.
So we're aboard the Nautilus with NASA subsea scientists and Woods Hole Oceanographic Institute scientists studying Gorda Ridge, the geological and microbiological organisms at the site. I encourage you to go to Nautilus Live and look at the expedition page, as well as all the other tabs there that may interest you.
So So we've got approximately another 4 minutes, is that correct, on this sample?
Yes.
Great, thank you.
When we have time, if someone, Trevor, could answer what's the pathway to become an ROV pilot?
Well, everyone has a different pathway. And it seems like every single person I work with has come from a different background, from technical schools to work experience to anything in between, being in the right place at the right time and taking advantage of opportunities when they arise. So myself, my way I got in there, the short answer is mechanical engineering. And the longer answer is a little bit longer than that, but it is kind of fun seeing why, like how there's no one defined path to get to be an ROV pilot. We work with people with PhDs, we work with people with college diplomas and master's degrees or trade schools or anything in between.
Thank you. And Ben, could you share about becoming a navigator?
Yeah, so I got involved in a lab in graduate school that is heavily interested in mapping operations, and whereas I was just more interested in other types of sensor information. But my interest kind of merged with being in that environment, and so I got involved through that connection with my lab.
Thank you. And Jessica, could you please share your role?
Sure, absolutely. So guys,
You're not an SPL, Jessica?
oh, thank you. Hi guys. So my name is Jess, and I'm the video engineering intern. And like Trevor said, my background is a little non-linear as well and non-traditional. I studied wildlife conservation biology in college. I have an undergraduate degree. But I was an artist. I almost went to art school, so I've always had a passion for, something visual and artistic, and so I managed to keep up filming and photography along the side and tried to figure out a way that I could combine those two passions. So I was trying to figure out how to combine filming with wildlife and took my camera with me wherever I went. And then I ended up volunteering and interning with our partners at the Inner Space Center, so I actually work for them as a production assistant, and I've been able to go out on different ships and film them and help connect them to the shore through telepresence. And I've learned a lot about that, and it's through the Inner Space Center that I learned about this internship. And so I came out here to learn more about the technology behind telepresence rather than just the camera work. So while I also help run and control the cameras for the science team and the ROV pilots, I'm also helping the IT department and make sure if anything goes wrong, we troubleshoot and make sure everything's working ship shape.
Thank you. Is there anyone else who would like to share their background?
Can we just get a quick temperature update?
50 degrees.
ICL's at 18.
Great, 50 and 18.
We're going to stop the filter soon.
Yeah.
Copy that, standing by.
Stopping sample now.
Confirm that, stopping 21:59:30.
Final flow count is 12420
12420
with a total volume of 6.66 liters.
6.66 liters.
An average flow rate of 0.33 liters per minute.
0.33 liters per minute. Copy that. Thanks, Eric.
Switching to next port.
Standing by. We confirm this is S6?
S6, correct. And I'm starting sampling right now.
Confirmed.
2230.
Starting 220030.
Starting flow counts the same as the ending.
Copy that, 12420. And temperature readings when you're ready.
I have 23 degrees right now.
15 on the ICL.
23 and 15. Thank you.
It's getting wafty.
Yeah.
Hey guys, can I take the eyes off the flow meter for a second?
Sure thing.
Thanks.
When the time's appropriate, Eric, could you share how you also have to engineer instruments, equipment to accommodate these expeditions?
Yeah, so putting things in the water might seem easy to a lot of people, but it really is not. And I'm sure the ROV pilots can commiserate that things can get damaged pretty quickly in the ocean. You need to protect it from seawater, number one, which is pretty bad for most electronics. And the fact that you have things moving around, they have to be protected from shock or smacking against things. So that super sampler inlet right there is actually 3D printed. It has an integrated PT100 RTD sensor, which senses temperature, and it uses resistance to do that. And because we have several different kinds of missions, like say microbiology, they want to see fluid samples around 20 to 70 degrees Celsius. We have to be able to know what temperature of the fluid that's coming out of that orifice while we're collecting it. And that's why you keep hearing us repeat temperatures for the data logger, is because we want to make sure that we stay within that temperature limit. The super sampler itself is essentially a rotating valve that allows us to select different passageways to collect that fluid and store it, whether it be running it through a filter or running into a bag or running into bottles to collect that fluid or to filter out microbiology that might be present in that fluid. And such we can collect that once we bring it back up to surface using the ROV. So a lot of ocean work that we do out here, you have to be able to adapt the technology to withstand the pressures of the ocean, as well as salt water and temperature changes, because we've seen even 300-degree vents at this site, and that would definitely melt any plastic that we have.
So you have to do quite a bit of research on material science as well as using CAD, etc.
Yeah, so we use CAD for 3D printing, and we can make sure the resins that we 3D print with can withstand the temperatures, or at least we know the max temperatures that can be used in. That way we avoid sticking into a 300-degree vent fluid and melting everything.
So if this was 300 degrees, that would be melting? Is that what you're saying?
Yeah.
All right.
Definitely. So would the super sampler.
Is it 300 degrees is the threshold?
No, the threshold is around 120 to 130, but that's a sustained burst of heat.
Yeah.
Like if we ran a little bit through there, it wouldn't melt right away, but you start having a little deformation.
Yeah.
And then over time, you'd have melty super sampler. Temperature, I have 14 degrees C.
ICL is at 31.
Copy that, thank you.
If you actually look at the tip of that super sampler inlet, the blue inlet, it's starting to get a little discolored. It's because we've actually stuck it into hotter temperatures than this the other day, but still held up.
A little toasted?
Yeah, a little toasty outside.
Thank you.
And do you have multiples of the wand, or is this your only wand? So if something was to happen there, it got in contact with maybe some super hot fluid. Are you able to?
I actually have a spare, but I try not to tell people that.
Uh-oh.
I don't want Trevor
Wait,
to
a spare?
No, Trevor, no spares.
How? What temperature does it melt at?
Let's test it.
I actually thought about bringing the 3D printer to sea with us. It would actually be pretty fun.
How would it work, do you think, at sea, like a 3D printer? Because obviously it like layers down the the resin or plastic. Do you think it would actually
we have actually two different types of 3D printer. We have the an SLA, which is stereolithography, and the other one is deposition printing, which is what you're talking about.
The layering.
Layering the filters.
Yeah.
Sorry, filament. And this was actually printed with stereolithography. What happens is it's in a vat of resin, and it uses UV light to cure each layer by layer. So you have very little porosity that forms in it because it's submerged in resin. However, the other one, fused deposition modeling, which is essentially you think about like a glue gun, layer by layer, that has more porosity. That will work perfectly fine at sea because it's mostly just a motorized table and arm. I have not tried the fused, the SLA version where it's in resin because obviously the resin is going to be rolling with the ship.
Yeah. What did that stand for again? SLA?
Stereolithography. I forgot what the A was, but it uses it uses UV light. It's actually pretty cool. It shines it layer by layer, and that helps cure each of the polymers to the previous polymers that were cured. So it aligns the polymers so they start locking in with each other.
Is that like the? Sorry, I'll let you do a temperature reading before I ask any more questions.
I got 36 degrees.
ICL is at 8.
That's great. Thanks, guys. So my other question really quickly. So I know your temperature probe is embedded, or not embedded, but is actually inside the blue wand. So to the right of that, what we're seeing is we've got one metal bar and then we've got one metal probe. Which one's actually the temperature or the thermocouple for the IGT ICL?
It's that
the
thinner metal wand to the right side. That's the temperature thermocouple from Sean's IGTs.
Okay, and what's the other bit? Is that just like a little brace for it, or is that part of
It's actually part of the handle that they use to secure things to so they can grab it.
Oh, okay. Okay, so that's like the hockey puck.
Yeah, the hockey puck has a little, I think it's aluminum rod coming out of it so they can secure like the super wand to it or thermocouples to it.
So to keep them as aligned as possible when they're actually taking the samples.
Yep.
Great, thank you.
This one's actually flowing pretty well. We're going to have a contest who has the better filter preparing strategies. I've got 71 degrees currently.
ICL is at 12.
71 and 12.
I bet it drops now.
I bet it does too.
I bet it does too.